Welcome to isolation of DNA with Rex. Today we are going to isolate the DNA from beetroot sample using CTAB method. The CTAB method consists of three important stages. One is the lysis, where the cells are lysed using the CTAB buffer and pestle mortar. Two, extraction using chloroform, where the proteins and lipid molecules are separated. Three, precipitation of the aqueous phase using isopropanol or ethanol. These are the materials required: CTAB, chloroform, isopropanol, and other minor requirements. So this is the composition of the CTAB buffer. This composition, it consists of tris as the major buffer, EDTA as the chelating agent, sodium chloride, CTAB as the detergent, and beta-mercaptoethanol as the reducing agent. We used 1 ml of this CTAB buffer for 100 mg of tissue. So weigh 100 mg of tissue and then homogenize in a pestle mortar using 1 ml of warm CTAB buffer preheated to 65 degrees Celsius. Later it is transferred to 1.5 ml or 2 ml centrifuge tube, and they are incubated at 65 degrees Celsius for 20 minutes in a water bath. So once 65 degrees Celsius heating is done for 20 minutes, they are transferred to a centrifuge, centrifuged at 10,000 rpm for 5 minutes. This step is to pellet the debris, the cell walls, unwanted macro molecules, and then transfer the lysate or the supernatant into a fresh tube. To this particular supernatant, we add equal volume of chloroform and mix by inverting it 5 to 10 minutes. So this will extract the proteins and lipid molecules from the lysate and provide a clear phase separation. One is the aqueous phase, which contains nucleic acid. The second is the solvent phase, which contains the chloroform. So this is done by centrifugation at 10,000 rpm for 10 minutes. Transfer the clear supernatant into a fresh tube and add equal volume of isopropanol or 2 volumes of ethanol. So isopropanol or ethanol will precipitate the DNA. So to enable the precipitation of DNA, we invert it by mixing the isopropanol and incubating it at room temperature for 20 minutes. It may be even incubated on ice or in a refrigerator for a longer time. So this will precipitate the nucleic acid. So once the nucleic acid is precipitated, you can centrifuge this particular tube at 10,000 rpm for 10 minutes, discard the supernatant, which is isopropanol, and wash the pellet using 100 microliter of 70% ethanol. The 70% ethanol contains 30% water, and this 30% water will leach or wash away the salts that co-precipitated with the DNA. So once the 70% ethanol wash is done, discard the supernatant, air dry the pellet to remove excess ethanol, and depending upon the size of the pellet, dissolve the DNA in 30 to 50 microliter of water. Load appropriate volume of DNA onto a 0.8% agarose gel and run it at 100 volts for 10 minutes. The DNA will appear once it's stained with ethylium bromide. It will appear on the top as indicated by an arrow. Since we did not treat it with RNases, RNA also co-precipitate with the DNA, which appears as a contamination which runs ahead of the DNA. Thank you.